Once the endomembrane system has created a lysosome, what would a cell do with a stomach, right? It doesn't have a mouth. What is it going to digest? So the next thing we need to talk about is what use is a lysosome for an animal cell. Well, there are certain types of cells that can engulf other cells or food particles using a process that's called phagocytosis. So remember, cyto stands for cell, and phage is to eat. This is essentially a way that cells can eat. The way that phagocytosis takes place is illustrated here in the top figure. So we have a cross-section through a cell. Here's the plasma membrane. Let's say the cell sees a piece of food on the outside. It detects it, right? It doesn't have eyes. It's going to detect that piece of food and says, I would like to take that in, and I would like to use that food particle as a source of macromolecules to build my own macromolecules. And so what the cell will do is it will allow its plasma membrane to pinch in. When that happens, it pulls that food particle into the inside, and when the plasma membrane completely pinches off, it's created essentially a vesicle that we call a food vacuole. This food vacuole contains the food particle in the middle of the membrane. With that, the cell by itself can't do anything. But what it does is it recruits one of its lysosomes to come over, and membranes can very easily fuse with each other. And so the lysosome membrane will fuse with the membrane of the food vacuole. This then allows the food particle to get access or be accessed by the hydrolytic enzymes in the lysosome. And all those hydrolytic enzymes will begin to degrade that food particle down to its smaller components, essentially digesting the food particle just like your stomach digests the food that you ingest. The cell can then absorb those different macromolecular components and use them as raw building material or fuel for the cell itself. Another way in which a cell can use a lysosome is in a process that's called autophagy. So again, phage is to eat, and auto meaning essentially self. So this is a way for the cell to eat itself, which seems kind of morbid. I like to think of it more like recycling inside of the cell. Cells are really good at being efficient. So for example, let's say there are organelles that have run their course and they're damaged and they're not functioning very well anymore. The cells, it doesn't have to keep them around. What it can do is it can take, let's say, this mitochondrion and this peroxisome that are no longer functioning very well. It can envelop them in a membrane. And if it envelops them in that membrane, it can then encourage a lysosome to come over, and the lysosome will go ahead and dump all of its hydrolytic enzymes into the membrane where the mitochondrion and the old peroxisome are, and those organelles are essentially completely degraded down to their macromolecular components, and the cell can use those components again to build organelles afresh. All right, so it's a way that cell ensures that all of its parts are working properly, and it can recycle those that are not working very well. All right, and so the last part that we need to discuss regarding the endomembrane system is that the endomembrane system can indirectly produce vacuoles. Vacuoles are essentially large vesicles that are derived from the ER and the Golgi membrane that have very specific functions. And there are a variety of different functions depending on what kind of vacuole you're looking at. So just on a previous slide, we already introduced food vacuoles, right? Food vacuole is that vacuole that's produced during the process of phagocytosis, where a cell takes in that food particle by allowing its plasma membrane to pinch in. We also can talk about, for example, contractile vacuoles. Now, contractile vacuoles, this will make a little bit more sense when we talk about osmosis and diffusion later on in Chapter 7. But there are some organisms that live in environments where they're constantly forced to take on water, like a ship with a hole in it, right? In order to stay afloat, you have to bail that water out. Same thing goes for these organisms that are constantly taking on water. They don't want to explode because they have too much water on the inside, and so they have to push some of that water out. So what they do is they collect that water inside of their contractile vacuoles, and as the name implies, that vacuole has the ability to contract and push all the excess water out, ensuring that the organism survives under those specific conditions. Okay, and then the last kind of vacuole that we're going to talk about is a central vacuole. Central vacuoles are only found in plant cells. So on the right-hand side here, you can see a cartoon diagram of a plant cell. And this sort of what looks like empty region in the middle is a central vacuole. Here's also an electron micrograph of a plant cell, and again, it looks like there's a big empty space in the inside of the cell. That's again the central vacuole. Central vacuoles, their main function is storage. They hold water and other organic, maybe compounds within the cell that the cell might need to use at some point.